Oh, it says your screen sharing is resume. This. Okay, can you guys see my screen? It's just like a thumbs up. Yes? Okay, perfect. All right, so we'll start with chapter 8. Chapter 8 is a pretty long chapter, and it's honestly more related to test 1 material. There's a lot of chemistry involved. So, chapter 8 is energy, entropy, equilibrium, and enzymes. So first, we have to start off with what potential energy and kinetic energy is. Potential energy is what most people refer to as like static energy. It's the energy stored within your chemical bonds. It's the energy found in concentration gradients, electrical gradients, and it's commonly referred to as your free energy that'll be eventually used up for kinetic energy. Kinetic energy, typically found in ATP hydrolysis. It's the movement of molecules down a concentration or electrical gradient. Radiant energy in light is considered kinetic energy, and heat as well. So there are two laws of thermodynamics. The first law is that energy can neither be created nor destroyed, commonly referred to as the law of conservation. So it's basically saying that in a closed system, energy will be transformed from one system to another or from one form to another. However, the total amount of energy before and after will still be the same. Now, the law of entropy, which is the second law of thermodynamics, states that entropy will never decrease. Entropy in this case is your unusable energy because, as we mentioned before, potential energy becomes kinetic energy. So entropy will never decrease, and entropy actually increases over time. Uh, sorry if you could mute up. Okay. Entropy actually increases over time, as shown by the unusable energy after increasing. So catabolism, anabolism, and metabolism. So for anabolic reactions are the reactions that just think of it as making things. Anabolic reactions link molecules together to make more complex molecules, and usually for anabolic reactions, you need an input of energy to create bonds. So energy is stored within the chemical bonds that are actually able to be formed. So anabolic is creation. Catabolism is breaking down. So you're breaking down complex molecules into simpler ones, and the actual breaking of the chemical bonds provides energy to be released. So energy efficiency, we have exergonic versus endergonic reactions. So for exergonic reactions, they release energy. So delta G, which is the amount of energy, is negative. So if you see in an exam, if you see a negative delta G and they ask what type of reaction it is, it will be an exergonic reaction. Exergonic reactions are associated with releasing free energy, and as a result, it is also spontaneous. So one thing students commonly get confused about is they think spontaneous means fast. Spontaneous, a spontaneous reaction does not necessarily mean a fast reaction. Spontaneous reaction just means that the reaction will occur without any outside input. So exergonic reactions have a negative delta G. They release energy and they're spontaneous, which means that they will occur without any outside input. Do you need to memorize the formula for delta G? For delta G, I think for this, just know it's the total energy of products minus the total energy of the reactants. That's pretty much all you need to know. So if you have lower energy products and higher energy reactants, obviously products minus reactants, your overall delta G would be negative, which means exergonic. And endergonic, your products have higher energy. Because your products have higher energy, your delta G will be positive and they consume energy. So endergonic reactions consume energy and it is non-spontaneous. Input is required so that the reaction can work. Equilibrium. So we'll talk about chemical equilibrium quickly. So you have something known as glucose 1 phosphate and glucose 6 phosphate. Glucose 1 phosphate is a very unstable molecule and it's required for glycolysis. And glucose 6 phosphate is a more stable version of glucose 1 phosphate because the phosphate near the carbon 1 has a lot of steric hindrance, but the phosphate on carbon 6 does not have a lot of steric hindrance, so it's a lot more stable. So as a result of that, what this slide is trying to say is that all reactions are reversible and an equilibrium exists. So in the original, in the glass or the jar on the left, you have 100% glucose 1 phosphate, but because it's so unstable, it wants to reach equilibrium. After it reaches equilibrium, we see that you have around 95% glucose 6 phosphate and around 5% glucose 1 phosphate. So all in equilibrium, in principle, all reactions are reversible. So glucose 1 phosphate can become glucose 6 phosphate, and glucose 6 phosphate can become glucose 1 phosphate. So cellular energy, I believe we talked about ATP in the first exam as well, but to those of you who aren't sure, ATP is essentially the currency of all cellular energy, and what it is you have an adenine bound to a sugar and then three phosphates. ATP stands for adenosine triphosphate, so three phosphates. And then some of the free energy released by exergonic reactions are captured by ATP, which can then release that energy to drive endergonic reactions. And an ATP can be hydrolyzed to yield ADP and an inorganic phosphate plus free energy. And what ADP stands for is adenosine diphosphate, so two phosphates. As you see here, you have ATP. It has one, two, three phosphates. ATPase can break it down, so you have ADP plus an inorganic phosphate here. And ADP has one, two phosphates. So the formation of ATP is endergonic reactions. So energy is required. So to form ATP, energy is actually required to bring a phosphate group sufficiently close to ADP for a bond to form. However, after hydrolysis, the energy stored within the phosphate oxygen here, the energy stored within this bond is greater than the energy required to bring it together. So as a result of that, free energy is actually released, and overall ATP hydrolysis is considered a exergonic reaction because more energy is released than energy required to bring the phosphates together. So this is what I was referring to. So ATP hydrolysis has a negative delta G, which means it releases energy. Here, the delta G is negative 7.3. And then eventually you can use that ATP or ADP to couple it with other reactions. So when you couple it with other reactions, you can add phosphates, etc. So in this case, you're adding a phosphate to glucose to convert it to glucose 6 phosphate. And even though adding a phosphate to glucose is a endergonic reaction because it requires energy, the overall delta G here is plus 4. So when you couple the reaction together, your net delta G is negative 3.3, which means that this is a overall exergonic reaction and it is spontaneous. Yeah. So, okay, I saw someone ask that they were a bit confused about this. Here, what I'm saying is that you can couple reactions together to make the overall reaction spontaneous. So for ATP hydrolysis, it is a exergonic reaction. However, adding a phosphate to glucose is a endergonic reaction. But when you couple it together, because you release more energy than energy required to combine the phosphate with glucose, the overall reaction has a net delta G of negative 3.3, and which means that it is a overall exergonic reaction that proceeds towards completion. So it is spontaneous. So we have what are known as catalysts. So catalysts help decrease the energy required for the activation energy. So some reaction rates are low because there is a large energy barrier between reactants and products. So actually having that reaction occur is very low. So and sometimes the catalysts help with that. Catalysts can help reduce that overall activation energy required so that the reactant can actually go and create the products. There is something known as a transition state intermediate. So the transition state intermediate is when a reactant obtains enough energy to go past the energy barrier. Once the reaction manages to go past the energy barrier, it becomes a transition state intermediate and it is very unstable. So once it reaches this transition state intermediate, it will 100% proceed with the reaction. But obviously, you need energy in order to be able to reach this little hill here. And so this is just showing what I was talking about. You have a stable state here, so the reactants are stable. But if you give it enough energy to make it reach that transition state, like you see here, then like that ball will naturally fall down. It'll be like that. As long as your reactant has enough energy to reach that transition state, it will proceed with the reaction to form the product. And so there are two ways to overcome this hill over here. One is by increasing the kinetic energy of the reactant, which is harder than expected, but the most common one is actually lowering this energy barrier. And the way to lower that energy barrier is, as we discussed, catalysts. Biological catalysts act by lowering the activation energy, and so enzymes do not change the delta G of the reactants or the products. So they don't change any of the overall final equilibrium. Enzymes only change the rate of the reaction. So enzymes just make sure the reaction will be able to proceed faster and you'll have more of it, more reactions occurring. Enzymes also help with the concentration of reactants and the concentration of products that'll occur. So as you see here, without an enzyme, it still goes up over time, but with an enzyme, you're able to reach the maximum rate much quicker because you're having more and more reactions, and it's easier to perform the reactions. So enzymes and their mechanisms. So substrates typically bind to enzymes. So substrates are reactants. They bind to enzymes at their active site and produce something known as an enzyme substrate complex. And this substrate enzyme complex is held together by hydrogen bonds, ionic interactions, van der Waals forces, etc. So an enzyme and a substrate bind, and eventually a product is released. So you have different types of enzymes. In a catabolic enzyme, an active site binds to one substrate to form an enzyme substrate complex, and in turn, it can release one or more products. So catabolic enzymes, you're binding to one substrate and you're releasing one or more products. In an anabolic enzyme, you're binding to multiple substrates to form the enzyme substrate complex, and you only release one product. And in this case, free energy is consumed. In catabolic enzymes, free energy is released, or you're releasing multiple products. But in anabolic enzymes, because you're binding to multiple substrates, free energy is consumed to release whatever product you're releasing. So enzymes allow for different orientations of the substrate. So naturally, they after binding with the substrate, they can help orient them in the proper positions or force them to overcome a specific repulsion. Another way is through inducing instability in the substrates themselves. So by binding with the substrate, enzymes can actually weaken bonds and make them more vulnerable to attack by hydrolysis, which changes obviously the structure of the substrate. Then there's also their own substrate modifications, where they can chemically modify the substrates after binding to it by adding charges, adding different functional groups. And then, as it says here, some of the interactions may involve covalent linkages between the enzymes and the substrates themselves. So there are multiple ways to create conformational changes within the actual substrate, and then the substrate also creates conformational changes within the enzymes. So the after binding with the enzyme changes shape so that obviously you don't want multiple substrates binding to the same active site. So the enzyme changes shapes. So there are some enzymes require partners with their substrates that they're bound to. So prosthetic groups are non-amino acid groups that are bound to the enzymes. As you see, you have several different types of prosthetic groups. Honestly, I don't think you'd need to memorize all the specific types of prosthetic groups or cofactors, coenzymes, etc. But I would say know what prosthetic groups are. So prosthetic groups, non-amino acid groups that are bound to enzymes. Inorganic cofactors are ions such as iron, copper, zinc that are permanently bound to the enzyme, help with oxidation, reduction, or stabilization. And then you have coenzymes, which are small carbon-containing molecules that are not permanently bound. They have their own roles and they can eventually unbind to the enzyme as well. Enzyme regulation mechanisms, as you see here, a lot of our metabolism of different amino acids, nucleotides, lipids, etc., everything is correlated with one another. So we have lots of regulation mechanisms to ensure we don't create too many substrates or to ensure we don't create too many products, too many substrates aren't binding, etc. So one way is irreversible inhibition. So typically what happens, an enzyme known as acetylcholinesterase here can hydrolyze acetylcholine into choline and acetic acid. However, there are molecules that can prevent binding of the active site, such as DIPF, which is also known as nerve gas. Nerve gas can form a covalent linkage with the serine residue here, and then it can inhibit irreversibly the active site. So it can make sure that after the original hydrolysis of acetylcholine, it can make sure that the acetylcholinesterase cannot bind to acetylcholine after. There's also something known as competitive inhibition. Competitive inhibition is when an inhibitor can bind to an enzyme's active site and it competes with the natural substrate for the site. So the substrate and the competitive inhibitor are actually competing to bind to the active site. But competitive inhibition can actually be overcome by increasing the concentration of the substrate. You can overcome competitive inhibition. There's also un and non-competitive inhibition. So in uncompetitive inhibition, an inhibitor binds to the enzyme substrate complex and it prevents the release of any products. In non-competitive inhibition, an inhibitor binds to an allosteric site. Allosteric site just means a site that's away or separate from the actual enzyme substrate complex. So an inhibitor can bind to the allosteric site, which changes the actual active site structure, preventing any binding. Here, so as you mentioned, some enzymes can assume multiple shapes. So when bound to an activator, as you see here, when bound to an allosteric activator, it can change shapes to become active. However, there are inhibitors as well. When bound to an allosteric inhibitor, they assume the inactive form. So they have different sites for activation, for allosteric activation and allosteric inhibition. So allosteric enzymes are controlled by allosteric regulators, which bind to a site that's separate from the active site. If this is the active site, the inhibitor binds here, the activator binds here, so they bind to a separate site. Positive allosteric regulators help stabilize the active form, while negative allosteric regulators stabilize the inactive form. Lastly, there's also, oh no, not lastly, sorry. There's also something known as feedback inhibition. So for feedback inhibition, your end product, by having an increased concentration of your end product, it can actually go back into the reaction and act as a non-competitive inhibitor to prevent any more formation of further enzymes. So feedback inhibition is extremely common. You'll see this in a lot of your body systems, which you'll learn about in your third and fourth exam. So feedback is commonly referred to as a negative feedback loop. So your final product acts as a non-competitive inhibitor of any of your original enzymes because there's an increased concentration. So it's essentially telling your body, hey, we have enough product. We don't need the substrates to keep binding to produce more of this product. So it shuts down the pathway. Then the last two regulators are pH and temperature. So pH can change the charges of the carboxyl and amino groups of the amino acids, which affects the interactions of the amino acids with the environment and helps change the structure of the protein. So as you see here, each enzyme is properly active at a particular pH. So for example, pepsin. Pepsin is extremely active at a pH of 2, which is extremely acidic. Why? Pepsin is commonly found in your stomach because it helps break down proteins in the food you eat. So your stomach obviously has a lot of acid, so pepsin works best there. However, chymotrypsin works better at a normal pH of around 7. Arganase works better at a basic pH. So enzymes work best at different pHs. And as you can see here, the reaction rates decrease significantly just by an increase of 1 to 2 pHs. Temperature as well is extremely important. So some organisms live at a very wide range of temperatures, and they help generate different forms of enzymes known as isozymes. But sorry if you could mute. But they function opt isozymes function optimally at distinct temperatures. And extremely high temperatures or extremely low temperatures can inactivate the enzymes by breaking non-covalent bonds. And as you mentioned, enzymes are proteins. So if the protein structure is disrupted, we refer to that as denatured. So high temperatures or low temperatures can denature the protein because it disrupted the structure of the enzyme, and so the enzyme would obviously not be able to work. So now we're looking at cellular respiration, chapter 9. So cellular respiration, you have two types. You have aerobic respiration and anaerobic respiration. Aerobic respiration is through the presence of oxygen. Anaerobic respiration, also known as fermentation, does not have oxygen. So cellular respiration, as you can imagine, because there is oxygen being used, your net energy per glucose is 32 ATP. So there's a lot of glucose molecules. So there's a lot of ATP molecules being produced. However, fermentation, because you're oxidizing uh, the glucose and pyruvate molecules incompletely, you're only producing around 2 ATP per glucose. And I believe you guys did a fermentation lab as well for in your lab class. So it's a similar process. Fermentation produces much less ATP, and you also see lactic acid formed. For example, when you're running and then you're running out of oxygen, you see you have lactic acid forming in like your legs, your muscles, etc. That's because your body does not have enough oxygen to perform cellular respiration. So lactic acid is being produced as a waste product, and you're only producing a minuscule amount of ATP. But first, we have to talk about what oxidation reduction is. So, oxidation is a loss of electrons. You're becoming more positive. Reduction is the gain of electrons. You're becoming more negative. And then here, students get very confused. So there's a reducing agent and an oxidizing agent. The reducing agent becomes oxidized. So the agent is what's causing the other molecule to become reduced or oxidized. So just when you think about what's a reducing agent, what's an oxidizing agent, think about what's helping the other molecule, like what's happening to the other molecule. So for example, for compound A, if the other molecule is gaining electrons, it is the reducing agent because it is giving up electrons. If the other molecule is losing electrons, then if the other molecule is gaining electrons, yes, oh my God. If the other molecule is losing electrons, as you see here in compound A, compound B would be your oxidizing agent because it is obtaining the electrons from compound A. So an oxidizing agent, as you see here, gains electrons and becomes reduced in the process. A reducing agent gives up electrons and becomes oxidized in the process. It says here, a lot of students think it's backwards. Even I got a little bit fumbled with my words as well. But just make sure to get through your head. Reducing agent becomes oxidized. Oxidizing agent becomes reduced. So now we're going to talk about glycolysis, which is the first step of cellular respiration. So glycolysis is the breakdown, also known as the oxidation of glucose to pyruvic acid or pyruvate. So as you see here, you have one molecule of glucose, and then two of the first three steps require ATP. So they're using energy from ATP hydrolysis in order to convert glucose into pyruvate. So glucose eventually gets converted into pyruvate. Oh, sorry, glucose gets converted to fructose 1,6 bisphosphate. So fructose 1,6 bisphosphate, it has two phosphates over here. So it gains two phosphates in the first carbon and then your sixth carbon over here. And then that fructose 1,6 bisphosphate can eventually be converted into two molecules of G3P. So essentially, the way to think about it, after producing fructose 1,6 bisphosphate, it gets cut in half, essentially. It gets cut in half, like right down the middle. So you have two molecules of G3P being produced. And then through further steps, which NAD plus gets NAD plus is an electron carrier that'll be used much later on in cellular respiration. But NADP plus gains the hydrogen, ADP and phosphates turn into ATP. So as you see here, you have four molecules of ATP being formed, and then eventually these four molecules of ATP being formed convert G3P into pyruvate. So you're not going to have to know each individual step. However, for glycolysis, you should know. You start with one molecule of glucose. That one molecule of glucose becomes fructose 1,6 bisphosphate thanks to two ATP molecules giving up their phosphates. That fructose 1,6 bisphosphate essentially gets cleaved in half so it can form two identical molecules of G3P. And then those G3P become pyruvate, and after becoming pyruvate, four ATP molecules are formed and two NADH molecules are formed. Um, NAD plus is an oxidizing agent in step 6. Correct. Because it is getting reduced. And so the first three steps, glucose uses two steps of ATP hydrolysis. Yes, exactly. So the first glucose converting into fructose 1,6 bisphosphate is three steps total. Out of those three steps, two of those steps requires ATP hydrolysis. And so here is a showing G3P becomes 1,3-BPG, 1,3-BPG becomes 3PG, but this is just showing more emphasis on how these NADP plus molecules are formed, how ADP gets converted into ATP. As you see here, NADP plus takes one of the hydrogens from the carbon here, and then in step 7, 1,3-BPG, ADP takes the phosphate from 1,3-BPG to form ATP, eventually getting you 3PG. So this is just showing how NADP plus and ADP, NADP plus is converted to NADH and ADP is converted to ATP. So we say that we have a net gain of 2 ATP from glycolysis because in the first three steps we use 2 ATPs, and then in the last four steps we formed 4 ATP. So there's a net gain of 2 ATP. And then we also formed two pyruvate molecules by the end. So now we're going to take a little break and talk about fermentation. So a little break from cellular respiration. Sorry. So now we're going to talk about fermentation. So fermentation is anaerobic respiration. So why do some cells use fermentation? So some cells use fermentation obviously because they don't have oxygen and they need to regenerate the NAD plus molecules to help keep glycolysis going. So fermentation uses pyruvate to oxidize NADH and regenerate NAD plus. And then that pyruvate is then reduced to lactate or ethanol. So after the pyruvate molecule is formed, usually it would go into pyruvate oxidation and then the citric acid cycle. However, without oxygen present, you still need to produce ATP because ATP is the main energy source for all of your cellular reactions, essentially. So the way they do that, they regenerate NAD plus using that pyruvate molecule that was their end product. So they regenerate NAD plus and it's a cycle again. Start at the beginning and then you're producing 2 ATP, that 2 pyruvate then gets regenerated back, etc. So for pyruvate oxidation, we have pyruvate, which was the end product of glycolysis. Pyruvate is then converted to acetyl-CoA using coenzyme A. Coenzyme A also allows NAD plus to obtain a hydrogen, and then carbon dioxide is released. That's essentially all you need to know about pyruvate oxidation. Pyruvate becomes acetyl-CoA through an enzyme known as coenzyme A, and NAD plus uh, obtains a hydrogen ion and becomes NADH. So now we're going into the Krebs cycle. So during the Krebs cycle, decarboxylation of each molecule of pyruvate helps yield one molecule of CO2 and acetate. That acetate is then acetylates coenzyme A to produce acetyl-CoA, and this acetyl-CoA will be responsible to be found in our citric acid cycle. So here you have acetate, also known as acetyl-CoA. Your in Another input is water, GDP. Your oxidized electron carriers, which are NAD plus FAD, and then your outputs will be carbon dioxide is released, reduced electron carriers because the NAD plus and FAD obtain electrons, and then a small amount of GTP is formed. So for the citric acid cycle, you won't need to know each individual step, what's happening, etc. The main thing you need to know is the inputs and the outputs. What is getting inputted? Acetyl-CoA from pyruvate oxidation, as well as water and some oxidized electron carriers here. Your outputs, carbon dioxide gets released. You have a very small amount of GTP, which can help transfer its phosphate to form ATP later on, but it's very small. But the main output of the Krebs cycle are these, your electron carriers. The electron carriers are vital for the last portion of cellular respiration to produce ATP. So this last portion is your electron transport chain. So oxidative phosphorylation, which helps form ATP. So NADH and FADH2, which you acquired from the citric acid cycle, are now electron carriers. So they carry electrons, and then you have something known as NADHQ reductase complex, which is your initial electron acceptor. So at the start of the electron transport chain, your reductase complex accepts electron from NADH, and then these electrons essentially get passed down. So there's a coupling of redox reactions that occur in each station. So each station oxidizes the previous station and becomes reduced. So the electrons get accepted in essentially in this case in a downwards fashion through each substation. So NADHQ reductase accepts the electron from NADH, passes it on to ubiquinone. Then you have succinate dehydrogenase. Succinate dehydrogenase accepts electrons from FADH2, which then sends it also to ubiquinone. Ubiquinone sends it to the reductase complex here, and it sends it to cytochrome C. Cytochrome C sends it to the oxidase complex, and your final electron acceptor is oxygen, forming water. So each station oxidizes the previous station by accepting the electrons. The electrons get passed down from station to station, and then oxygen is your final electron acceptor, becoming water. So what's the point of accepting these electrons? The main point of accepting these electrons is to form a proton gradient. So as electrons are accepted, protons travel through the proteins. So protons travel through each complex, forming a proton gradient on one side. So hydrogen ions are going through as each electron gets accepted. So now you have a huge proton gradient here. And so this creates an electrochemical charge across the membrane through H plus transport. So here in the inner membrane space, you have extremely high hydrogen ion concentration and extremely positive charge. However, in the actual matrix of the mitochondria, you have low hydrogen ion concentration and a negative charge. So this proton gradient is extremely important because in order to form ATP, your hydrogen ions go through what's known as ATP synthase. As your hydrogen ions go through ATP synthase, your F0 unit is kind of like a motor, which just circles around, and then your F1 unit helps convert ADP into ATP by combining ADP and a phosphate together. So electrons get accepted by these complexes here and get passed down. Eventually, oxygen is your final electron acceptor, producing water. The point of accepting all these electrons and passing it down is to create this proton gradient right here. As the electrons get accepted, protons go from the matrix of the mitochondria to the inner membrane space, creating a proton gradient. You want the protons to go down its gradient to maintain equilibrium, so the protons go down, ATP synthase, the F0 unit is like a motor rotating, and then this helps combine ADP and phosphate, producing ATP. And so this last step helps produce around 30 to 32 ATP. So the hydrogen ions enters, so how does that help with ATP production? So the hydrogen ions going down the ATP synthase essentially binds to ADP and phosphates. There are free roaming phosphates rolling around. Hydrogen ions going down the ATP synthase acts acts like a motor. You kind of think of it as like this if you think of ATP synthase as a car, hydrogen ion is your gas, helping pump your car so that the car can take you to your destination. In this case, the destination is converting ADP into ATP by combining it with a phosphate. Hydrogen ions act as the energy source for ATP synthase to rotate and move, combining ADP and phosphate together. And oh yeah, you could think of it like a river pushing a wheel, exactly. So yeah, the wheel helps combine ADP and phosphate, and that river would be your hydrogen ions going down its concentration gradient. All right, so now we're going into photosynthesis. Photosynthesis is what typically people refer to as kind of like the opposite of cellular respiration, because in photosynthesis, we are producing sugar instead of producing ATP, while in cellular respiration, we used sugar to produce ATP. So in photosynthesis, you have two pathways, the light reactions and then the Calvin-Benson cycle, also known as your light independent reactions. So during the light-dependent reactions, you have solar energy being converted into chemical energy, and it's driven by light energy captured by chlorophyll. It consumes water, produces oxygen, ATP, and NADPH. But your Calvin-Benson cycle does not actually use the light directly. It uses the products of the light reaction in order to eventually produce sugar. So both pathways rely on each other and then depend on each other because the Calvin-Benson cycle uses the products of the light reaction to help produce the sugar required for the plant. So this is just describing the plant cell. You have the cell wall, vacuole, etc. But the main spot we're going to look at is the chloroplast. The chloroplast is where photosynthesis takes place. So within the chloroplast, you have something known as the thylakoid and the stroma. So the thylakoid are like these granules stacked on top of each other, which is where the light dependent reactions happen. Your light dependent reactions happen in the thylakoids because your thylakoids are where your chlorophyll is located, and the chlorophyll is able to absorb light from um obviously the environment. And so the stroma is the fluid surrounding the thylakoid. So you kind of think of the stroma as like the cytosol of the chloroplast. Within the stroma is where the light independent reaction or the Calvin cycle occurs. So light dependent reaction occurs in the thylakoid of the chloroplast, and then light independent reactions occur in the stroma of the chloroplast. So now we're going to talk about the light reactions. So one way that the light reactions occur is non-cyclic linear transport. So for non-cyclic linear transport, you have photosystem 2 and photosystem 1. So I remember a few years ago, there was a question on the exam that confused people because they asked, what is the order of how do photons move through the light in a light dependent reactions? And so the correct answer would be photosystem 2, electron transport chain, photosystem 1, NADP plus reductase. So that would be the correct answer. However, a lot of students chose the wrong answer. They chose photosystem 1, comma photosystem 2, electron transport, etc. So the reason they chose photosystem 1 first is obviously if it's labeled photosystem 1, you would think that that would be the first uh proton acceptor or electron acceptor in the sequence. However, photosystem 1 is not the primary acceptor. The reason it's labeled that way is because photosystem 1 was discovered earlier than photosystem 2. Photosystem 2 was discovered years after photosystem 1, and obviously they can't change the names around, so that's why in the non-cyclic electron transport, we start with photosystem 2. And even in cyclic electron transport, we start with photosystem 2. Photosystem 2 is your initial acceptor. So what happens? You have chlorophyll molecules here. Those chlorophyll molecules become excited thanks to light. So chlorophyll molecules absorb light and they excite electrons. These electrons are obtained from water being broken down into oxygen and hydrogen. And so this water being broken down into oxygen is what obviously provides humans the ability to breathe because plants provide oxygen to the environment through this process right here, where they break down water into oxygen and hydrogen. And then the electrons from H2O get excited because chlorophyll absorbed light. As the electrons get excited, they go through the electron transport chain. The electron transport chain is actually relatively similar to the electron transport chain you see during ATP production. As the electrons go through the electron transport chain, ATP is formed. But the electrons' jobs aren't done yet. Photosystem 1 chlorophyll accepts the electron, then the electron gets excited again. Then these electrons go through what's known as ferredoxin. Ferredoxin, then the electrons go through ferredoxin, then NADP plus reductase, which then combines NADP plus and hydrogen ions to form NADPH, which will be an electron carrier. So the main things you're trying to form in your light dependent reactions are ATP as well as your electron carriers. And then oxygen is a product as well. So once again, photosystem 2 into your electron transfer chain, into photosystem 1, into ferredoxin, NADP plus reductase will be your sequence if they ask about it on the exam. And so Okay, here's just describing that again. You have water being broken down into hydrogen ions and oxygen. Photosystem 2 obtains energy from a photon. Photon is your light energy. As it obtains energy from a photon, an electron gets excited. It goes through it goes through different complexes. These complexes obviously cause hydrogen ions to go through as well. As the electrons go through different complexes, they eventually end up at NADP plus reductase, where NADP plus becomes NADPH, an electron carrier. The hydrogen ions that are now in your stroma, the hydrogen ions that are now in your thylakoid go through your ATP synthase into your stroma, producing ATP. Okay. So which sequence gives a correct order of steps taking place during the light reaction? I'll give you guys like 30 seconds. And then you could direct message me if you don't want your answer being seen in the chat. All right. We have a few different answers. The correct answer is D. So light energy, light energy is required for chlorophyll to excite the electron. So light energy is required for chlorophyll to excite the electron. That electron goes through the electron transfer chain. Electron transfer chain creates a gradient, which then is responsible for producing ATP synthesis at the end. Also, I know in your slides there's something known as chlorophyll A versus chlorophyll B. So chlorophyll A is the main chlorophyll that excites the electrons. So it's the main pigment required for absorbing light. Chlorophyll B acts as an accessory pigment. So chlorophyll B acts as an accessory pigment to help chlorophyll A absorb more light and then excite more electrons. So someone asked why the answer isn't A. The reason the answer isn't A is because you can't produce ATP synthesis without obviously the electron transfer chain, and you can't produce a gradient without the electron transfer chain. So before everything occurs, the electron transfer chain has to come first in order to create the gradient so that ATP can be synthesized. What does the electron transfer chain provide the Calvin cycle with? A Calvin cycle, also known as your light independent reactions. So definitely know the two names because they can ask Calvin cycle, Calvin-Benson cycle, light independent reactions, etc. They could ask any name of those. And then 30 seconds for this as well. All right. Looks like yeah. Okay, a lot of people got it correct. The correct answer is B, ATP and NADPH. I saw someone said, see, the reason it's not water and photons are light energy, so they're absorbing photons. And the reason it's not water is because water is used to produce oxygen. So water is actually broken down to produce oxygen. But the correct answer is ATP, which is obviously energy, and NADPH, which is responsible for being the electron carriers for your Calvin-Benson cycle. So in your Calvin-Benson cycle, now we're going to what the actual reaction products are, which is glucose plus more oxygen plus more water. So the Calvin-Benson cycle uses carbon dioxide from the atmosphere in order to help produce the glucose at the very end. And this process is also known as carbon fixation. So first we have to talk about how this process was even found out. So this process was found out through the Calvin-Benson experiments. So cholera is a unicellular freshwater green algae which help provide the photosynthetic system. So exposure of chlorella cells to carbon dioxide for various durations resulted in compounds labeled with 14C, which is an isotope. So one of the first stable compounds they found that was labeled with it was 3-phosphoglycerate, also known as 3PG. And so other products of the cycle were found by increasing the length of time of exposure to carbon dioxide. As they increased the length of exposure to carbon dioxide, they managed to find more and more products until eventually the whole pathway was revealed. And so this is your Calvin-Benson cycle. Your products are 2G3P, which eventually becomes sugars, 18 ADP, and 12 NADP plus. And so the Calvin-Benson cycle is also has another name, the reductive pentose phosphate cycle. So it takes carbon dioxide from the atmosphere to produce carbohydrate. This process is known as carbon fixation. So the initial step is carbon fixation, which carbon dioxide is converted into a 5-carbon compound known as ribulose 1,5 bisphosphate, also commonly known as RuBP. So this reaction is catalyzed by a protein known as rubisco. And rubisco is actually the most abundant protein on Earth. So once again, initial reaction, you take carbon dioxide from the atmosphere, rubisco converts carbon dioxide into RuBP. However, RuBP is very unstable. Because of RuBP's instability, because of RuBP's instability, it eventually creates another unstable 6-carbon intermediate that breaks down to form down 3-carbon 3PGs. So 1, 2, 3, 3-carbon 3PGs. So 6 RuBP become 12 3PGs. So RuBP takes a carbon from carbon dioxide, becomes 6-carbon, and then essentially gets cleaved in half to produce 3PG. Now, after producing 3PG, ATP gets hydrolyzed into ADP, NADPH loses its electrons, and your 3PG become G3P. One out of your six, or in this case, two out of your 12 G3P become your sugars. So these sugars don't have to be glucose, it could be cellulose, it could be starch, but 2 out of the 12 G3P become sugars. However, the remaining 10 G3P need to regenerate RuBP. So the way that G3P gets regenerated, G3P eventually becomes RuMP. After that RuMP is formed, 6 more ATP is used and converted into ADP, and RuMP becomes RuBP, starting the cycle again. So one thing a lot of students get confused about is why do why does the plant require so much G3P converting back into RuBP? Why doesn't it convert all of it to sugars? And the reason is you can't even start the Calvin-Benson cycle process without having your starting material. It's similar to fermentation where that pyruvate converts NADPH back into NAD plus so that the process can start again. You need the starting material in order to start the cycle. If it converts all of it into sugars, there won't be any starting material left, so the process or the cycle won't be able to occur. So as a result of that, the plant cycle allows most of the G3P to remain within the cycle to help regenerate RuBP. So for this, what you're required to know, obviously there is a lot of compounds, there is a lot of molecules, but you will be required to know one, two, three, four, what happens here, five, six, like these 6 steps. So once again, carbon dioxide combines with RuBP through a process through a rubisco, a protein known as rubisco, which helps catalyze it. This then creates an unstable intermediate. Unstable intermediate gets cut in half to produce 3PG. 3PG becomes G3P. 2 out of 12 or also 1 out of 6. So on the exam, they can use 2 out of 12, but 2 out of 12 as a fraction can be simplified to 1 out of 6. So they can say 1 out of the 6 G3P becomes sugars. So 1 out of 6 G3P becomes sugars. The remaining 5, or in this case 10 out of 12, the remaining 10 go back into the cycle to produce RuMP, which then becomes RuBP again. All right, knowledge check. Which of the following occurs during the light independent reactions? All right. Well, yeah, everyone got it correct. Carbon dioxide is converted into sugars. This was fairly simple. So now we're getting into the genetics portion of the class. So first, we're going to talk about mitosis and meiosis. So the cell cycle and cell division. So why does cell division occur? So three main reasons are reproduction, growth, and regeneration. So obviously reproduction is a lot of unicellular organisms reproduce through mitosis. Uh, there are several cell divisions which help contribute to the growth of different plant tissues and obviously human tissues as well. And especially in multicellular organisms, we focus more on the growth and regeneration aspect of it. So the general steps for cell division, you have a reproductive signal to initiate it, replication of your DNA, segregation, which is the distribution of DNA to the two new cells, commonly referred to as mitosis or meiosis, if we're talking about gametes, and cytokinesis, which is the separation of your two new cells. So initially, we're going to talk about prokaryotic cell division. Prokaryotic cell division is commonly referred to as binary fission. So, what are the factors that initiate it? So, external factors such as nutrient concentration, environmental conditions are the reproductive signals that initiate cell division in prokaryotes. So, for lots of bacteria, abundant food supplies help speed up the division cycle. So, as you know, if you're in a petri dish, if you give certain cells a lot of food supplies, they'll be able to reproduce abundantly. So that's their initiating factor. And how binary fission occurs, you have a plasmid in the middle. So a plasmid is just a small circular DNA molecule. So DNA replication begins at the orrery, which is at the very center of the cell. So the DNA replicates as the cell grows in size because it has to store that DNA while it's replicating. Then the daughter DNA separates, and as the daughter DNA separates, cytokinesis happens, which just cuts the cell in the middle in two. So now you have two new cells that are identical to each other. So prokaryotic cell division is known as binary fission. Oh, I you guys don't need to answer it yet. I just said it. This is D, binary fission. Okay, this is a good question. This has some test one material as well. All right, yeah, the correct answer is A. So bacteria, very simple. They have one chromosome that is circular, that is commonly referred to as a plasmid. Eukaryotes have many chromosomes that are linear. So great job. Now we're going to talk about eukaryotic cell division. So before we get into mitosis, we have to go through what's known as interphase because most cells actually spend the majority of their time in interphase. Mitosis is only a small portion of it. So in here, they only show three parts of interface, but there's actually four. There's something known as G0. So G0 is known as the cell arrested phase. If a cell is not destined to divide any further, it usually leaves the cell cycle and it stays in G0. Until it gets a signal to divide, most cells stay in G0. However, within interphase, you have G1, S, and G2, which is all about preparation for cell division. So after receiving a signal to divide, in the G1 phase, your chromosomes are single unreplicated structures, and then in G1, your organelles start to reproduce. So for example, your proteins, your Golgi apparatuses, etc. So your organelles, important organelles, start to reproduce, or not reproduce, start to duplicate itself. Then in the S phase is when your DNA replicates itself. So G1 is your organelles replicating, S phase is your DNA replicating. Then your G2 phase is just all about preparation for mitosis. So synthesizing microtubules to help move your chromosomes, etc. G1 organelles, S is your DNA, G2 is just preparation. Okay. Another knowledge check. All right, correct. Yeah, everyone got it correct. The answer is A. So the reason the answer is A is because G2 occurs after the S phase, as you see here, G2 occurs after the S phase where the DNA has replicated. As a result, the G2 nucleus has doubled the amount of DNA. So the reason it's not D is because during G2, the cell prepares for mitosis. During if the It's during G1 that the cell prepares for S phase. But during G2, the cell prepares for mitosis. And now, what happens in mitosis? So now you have prophase, prometaphase, metaphase, anaphase, telophase, commonly referred to as PMAT. So during prophase, your chromosomes, so as you hear, your chromosomes are kind of like loose. It's just a bunch of DNA all unwoven. But during prophase, your chromosomes start to become visible and they start to intertwine with one another. They start to condense. And your centrosomes start to move to opposite poles. And during prometaphase, the nuclear envelope, so this nuclear envelope, which was holding the DNA, the nuclear envelope starts to break down, and your kinetochore microtubules start to appear. And they connect the kinetochores to different poles. So they connect your kinetochores to the opposite poles of the cell. And then your chromosomes are gradually pushed together towards the equatorial plate. So once again, prophase, chromosome DNA starts to condense so that you can actually see your chromosomes. Your nuclear envelope starts to break down. Kinetochore microtubules, which is responsible for pulling your chromosomes apart, start to form, and your kinetochores go to opposite poles of the cell so that division can be equal. Metaphase, all that happens, your chromosomes line up at the equatorial plate or the M plate. They just line up in the middle. Anaphase is the actual pulling apart. Your sister chromatids separate. So chromosomes are made up of two sister chromatids. The sister chromatids separate and they get pulled apart to each end or each pole of the cell. Lastly, telophase is the opposite of prophase. In telophase, the nuclear envelope starts to reform, your kinetochore microtubules start to disappear, and the chromosome starts to decondense. And lastly is cytokinesis, which is it gets split in the middle, and now you have two identical daughter cells. So prophase, metaphase, anaphase, telophase. Prophase and telophase are the opposite of each other. Metaphase meets in the middle. Anaphase is pulling apart. Um, I'll skip this question because I went over it. So the correct answer is A, two genetically identical daughter cells are formed. So more about the microtubules and the kinetochore microtubules. So you have polar microtubules, which extend from the two spindle poles. So here, you have polar microtubules which extend from the two spindle poles and they overlap in the center. And then what they do is they help keep your poles apart. So they help make sure that it's on. You have your poles on. Sorry if you mind muting. Um, Zoom user. Um, let's see, how do I? Oh, okay, thank you. Um, oh, here. So you have your polar microtubules. So they extend from the two spindle poles, overlap in the center, and they help make sure the poles are on opposite side of the cells. Then you have your kinetochore microtubules. Your kinetochore microtubules are important because they attach to kinetochores on the chromatids, and after they attach to kinetochores on the chromatids and the spindle poles, during anaphase, that's what actually results in the breaking apart of the sister chromatids so that you have one sister chromatid, you have one portion on one side, the other portion on the other side. So chromatid attachment and separation. So at during prophase, the sister chromatids are held together by a molecule known as cohesion. In pro-metaphase, most of the cohesion is removed except for that at the centromere. So at the end of metaphase, you have a cyclin CDK complex. So that activates a APC. APC stands for anaphase promoting complex. That anaphase promoting complex activates an enzyme known as separase. Separase hydrolyzes cohesion and then allows obviously the removal of cohesion so that the sister chromatids can actually separate apart from each other. So during metaphase, at the end of metaphase, beginning of anaphase, APC, which stands for anaphase promoting complex, gets activated. Enzyme known as separase gets activated. Separase then gets rid of any remaining cohesion, which is located at the centromere of the cell, allowing the sister chromatids to separate from each other. So now we're going to talk about meiosis. So meiosis is the production of 1N haploid gametes by 2N diploid cells. So what does 1N and 2N mean? 2N is the typical amount of DNA found in the cell. So we commonly refer to those cells as diploid. If it's diploid cells, it's 2N. That's the normal amount of DNA. Haploid cells are half the DNA. They're 1N. And the haploid cells that are typically in meiosis are your egg cells in females, sperm cells in males. Those are your gametes. So meiosis is specifically the production of your gametes by diploid cells. Meiosis consists of two nuclear divisions instead of one. Meiosis consists of meiosis 1 and meiosis 2 to help reduce the chromosome number. DNA replication occurs only in the very beginning. DNA replication does not occur again after meiosis 1. It occurs before meiosis 1, so that after the two divisions, you have half the DNA in each of the daughter cells. And you have four daughter cells instead of two, and the daughter cells are different from the parent cells. Another thing, the reason why you need 1N or half the DNA in your gametes is because when the egg and sperm fuse to create a zygote, their DNA combines. So if their DNA combines, 1N plus 1N gets you 2N, the zygote has the normal amount of DNA. If your gametes were 2N each, then the zygote would have double the amount of DNA, which is not feasible. So that's why your gametes have to be 1N. That's why you need two divisions so that when the zygote forms, okay, can you explain a little bit slower? Yeah, sorry. Um, all right. So your gametes are your egg and sperm cell, which have half the DNA of all your cells. Your egg and sperm cell fuse to form a zygote. The zygote has 2N DNA now because 1N plus 1N from the egg and the sperm cells, after they fuse, they combine their DNA. Now you have the normal amount of DNA in the zygote. If the haploid gametes were diploid, so if the gametes were 2N each, your zygote would have double the DNA of the normal cell, which is not feasible. That's why you need two divisions in meiosis to make sure that your daughter cells have half the DNA. So, unlike mitosis, in meiosis 1, your homologous pair of chromosomes are paired together at the metaphase plate. Whereas in mitosis, they're in a straight line. They line up independently at the metaphase plate. Another difference is obviously there's two divisions in meiosis compared to one in mitosis. And there's also something known as crossing over, which we'll go over a bit later. So, phases of meiosis. The good thing about meiosis is that it is exactly the same thing as mitosis, just twice in terms of what actually happens within the cell. You have prophase, which is your DNA starts to condense into chromosomes. Your centrosomes start to go on opposite sides of the poles, and kinetochore microtubules start to form. The prometaphase, nuclear envelope starts to break down, and like I mentioned, chromosomes condense. Then in metaphase, your chromosomes line up at the equatorial plate or the middle of the cell. Anaphase, your chromosomes split apart. Telophase is the opposite. Chromosomes start to decondense, nuclear envelope reforms, cytokinesis occurs. And the exact same thing happens again. Now you have prophase 2, metaphase 2, anaphase 2, telophase 2, and lastly, you have four daughter cells, each that are genetically different from each other. So, as mentioned before, DNA does not replicate before meiosis 2, and in meiosis 2, sister chromatids will not be identical because of crossing over. So, if we look here, in late prophase, prometaphase, there's something known as the chiasma. The chiasma is the site of crossing over. And as you see here, you have your red chromosomes and your blue chromosomes. But now, some of the red chromosomes have some of the blue chromosomes, some of the blue chromosomes have some of the red chromosomes, etc. This is known as crossing over, where your maternal and paternal chromosomes exchange DNA segments with each other. Because they exchange DNA segments with each other, it allows for genetic variety, and this is what allows you to have four completely different daughter cells rather than identical daughter cells. It is specifically because of this crossing over, which allows DNA exchange segments between your chromosomes. Okay. So there are fertilization and meiosis alternates in sexual reproduction. So crossing over occurs in prometaphase is where I would say. Oh yeah, so there are fertilization and meiosis alternates in sexual reproduction. So certain fungi have a haploid mature organism, and only the zygote is diploid. So interestingly enough, the zygote is diploid, but then the zygote undergoes meiosis, and then your mature organism is haploid. Then you have alternation of generations. So in alternation of generations, the organism passes through both life cycles. So it has a haploid life cycle and it has a diploid life cycle. It decides it based on the environment. So in the diplontic life cycle, the organism itself is diploid, the gametes are haploid, which is in humans, most animals. So typically in fungi, you'll see the haplontic life cycle where the mature organism has half the DNA, and then you have certain ferns, plants, etc. that can alternate generations based on environmental conditions. Um, yeah, knowledge check. Okay, we have a few different answers. But the correct answer is D, produces less variation among offspring. So there is a major drawback with asexual reproduction. Although asexual reproduction is very quick, such as in bacteria and binary fission, because it is very quick, the issue is that there is no genetic variation. So if for some reason there's an environmental condition that will cause the death of one of the cells, all of the cells will die off unless there's a mutation, all of the cells will die off because there's no genetic variation from crossing over. So the correct answer is D. Asexual reproduction produces less variation among offspring. So in meiosis, because of how many processes are involved in meiosis, from prophase, metaphase, anaphase, telophase one to the second process of going through everything again, there can be meiotic errors. So meiotic errors are commonly referred to as non-disjunction. Non-disjunction is when your homologous pairs fail to separate or your sister chromatids fail to separate. So this results in aneuploidy. Aneuploidy means you have less chromosomes or greater chromosomes for certain uh, chromosome numbers. So you can have a monosomy, which is one less chromosome than expected, a trisomy, which is one greater chromosome than expected. So as you see here, um, during meiosis one, it splits apart. However, during meiosis one, it splits apart. However, non-disjunction occurred. So now you have aneuploidy, where one of your daughter cells has essentially none of the chromosomes. Another of your daughter cells has greater chromosomes, an extra chromosome. So after fertilization of that cell, you see in the monosomy you have half the chromosomes. In a trisomy, you have an extra chromosome. So in humans, a common uh trisomic condition is chromosome 21, or if in chromosome 21 homologs go to the same pole and the egg is fertilized, it will be considered trisomic for chromosome 21. This results in what is known as Down syndrome. So here's the thing. Meiotic errors are actually much more common than people expect. So trisomies and monosomies are actually pretty common in human zygotes. However, why don't we see more um why don't we see more actual symptoms of it other than, for example, chromosome 21 and Down syndrome? So the reason is most trisomies and monosomies are actually lethal. So they result in miscarriages. As a result of that, only very few trisomies and monosomies in very specific chromosomes will actually be able to have a zygote come to term and produce a baby which will be able to live. Most of them result in a miscarriage within the womb itself. Okay, so I just answered this. So yeah, most chromosome abnormalities are not observed because they are lethal and cause spontaneous abortion within the embryo early in development. So now we're going to talk about chapter 12, genetics. Genetics is one of the chapters that a lot of students struggle with, so I'll take a bit of time with this one. Um, just give me a second to drink some water. All right, so chapter 12, inheritance. So we have genes and we have alleles. So what are genes? What are alleles? So genes are coding for specific characteristic traits of the human body. And then alleles make up genes. You have two alleles per gene. You have you can have heterozygous alleles or homozygous alleles. So as mentioned, you have two alleles per gene. Genes code for specific traits. Heterozygous alleles is when you have the same gene at the same locus, but they carry different alleles. So heterozygous alleles would mean you have one dominant allele and one recessive allele. In a homozygous allele, same gene at the same locus. Locus is just the location on the actual chromosome of the gene itself, but they carry the same alleles. So you have either two dominant alleles or two recessive alleles. So once again, heterozygous, one of each. Homozygous, either two dominant or two recessive. So dominant alleles are your alleles that are expressed in an organism preferentially. Your recessive allele is the allele that is either not expressed or weakly expressed. And dominant, if you have both the dominant allele and the recessive allele, your dominant allele will be the one that's preferentially expressed in the organism. So here are just some examples of dominant genes versus recessive genes. For these dominant genes, you can have either both of the dominant alleles or having just one of the dominant allele is fine. In the recessive gene, you need two recessive alleles in order to actually express it. So for blonde hair, you need two recessive alleles for blonde hair. For the attached earlobe, two of them. Versus for brown black hair, you only need one of the dominant allele. So now genotype versus phenotype. Genotype is the genetic makeup of the actual individual. So when they ask for the genotype, they ask for the combination of alleles. Your phenotype is the physical expression of the individual. So what is actually physically seen. And organisms that have the same phenotype can have different genotypes. So if we say brown hair and versus blonde hair, the genotype would be uppercase H, lowercase h, uppercase H, lowercase h. That's your genotype, the combination of alleles. Your phenotype would be, do they have brown hair? Do they have blonde hair? And then organisms that have the same phenotype, as you mentioned, can have the different genotype because heterozygous and homozygous dominant showcase the same phenotype. So now we're going to get into crosses. So a true breeding cross is for traits that is the only one present for many generations. So true breeding traits are homozygous. So in this case, you have a homozygous dominant parent breeding with a homozygous recessive parent. So uppercase A, lowercase a. As you see here, all of the children have the same exact genotype and the same exact phenotype. They all have the genotype of uppercase A, lowercase a, so they're heterozygous for both traits, and they have the same phenotype as well. And then when you self-pollinate, so if you combine two heterozygous parents, you have what's known as the 1 to 2 to 1 ratio or the 3 to 1 ratio. I would recommend memorizing this specific ratio. So when monohybrids self-pollinate to produce the second generation, it's known as a monohybrid cross. And so the 1 to 2 to 1 ratio is the genotype ratio. It refers to the ratio of homozygous dominant, homozygous recessive, and heterozygous. So in this case, you have one homozygous dominant, two heterozygous, and one homozygous recessive. But your phenotypic ratio is 3 to 1. You have three dominant, one recessive. So now we're going to talk about how monohybrid crosses, Mendel's crosses came about. So Mendel used peas. And so he took peas with completely contrasting traits. He took a true breeding round seed, so a seed that's dominant for both, that's homozygous dominant for that round trait. And then he took a true breeding wrinkled seed, so a seed that's homozygous recessive for that wrinkled trait. After pollinating it, he found that all the F1 generation produced children that are all round. However, as we mentioned previously, they are all heterozygous. And then when you self-pollinate all heterozygous children, when you self-pollinated that plant, you have your F2 seed generation. In your F2 seed generation, you have, as you see here, three dominant, so three round, one wrinkled, which, as we mentioned before, is your 3 to 1 ratio for phenotype, 1 to 2 to 1 ratio for your genotype. Oh yeah, and as it says here, he was trying to see if recessive traits can reappear in future generations or if they just go away if you made it with a dominant allele. So as you can see here, recessive traits can appear in future generations, even if in one of the generations it's not shown at all. So this is just showing the cross. There are two alleles for P texture, uppercase S for round, lowercase s for wrinkled. After the F1 generation are heterozygous for both parents. After combining two heterozygous parents together, you have your 1 to 2 to 1 ratio. So the Punnett square is the main way people use to predict and display allele combinations. So now we're going to talk about the law of segregation. The law of segregation states that two alleles that code for the same trait separate during the formation of gametes. So you won't have two dominant alleles on one side. The alleles themselves will separate so that one gamete will contain one allele, the other gamete will contain the other allele. That's it for the law of segregation. Now we're going to talk about dihybrid crosses. A lot of students struggle with dihybrid crosses, and typically you won't need to actually make a dihybrid cross yourself, but on the off chance you do, I'll explain how to actually make a dihybrid cross. So what is a dihybrid cross? A dihybrid cross is a cross to find a generation, but you want to look at two traits rather than one. If you're looking at two traits, you're looking at two genes. If you're looking at two genes, you're looking at four alleles because two alleles per gene. So you're looking at four alleles total of one parent, four alleles total of another parent. 4 times 4 is 16. You will have 16 possible combinations. So how to actually create your dihybrid cross. So I refer to it as like a claw method. Some people just are able to figure it out on their own. But the way to actually figure out how do I set up the dihybrid cross, like combining everything is simple once you set it up. But a lot of students struggle with setting it up. The way to actually set it up is what I refer to as the claw method. So each allele can bind to any of the other two alleles of the other gene. Remember, that's what we mentioned about law of segregation, law of independent assortment as well. So this uppercase S can bind with this uppercase Y or this uppercase Y. If you look at this, the heterozygous, it's a bit easier to explain. This uppercase S can be bound to this uppercase Y, but can also be bound to this lowercase y. So you essentially create lines from each gene. So this S create a line with this uppercase Y, this S create a line with this lowercase y. So this S is satisfied. However, the lowercase s is left. So create a line from the lowercase s to the uppercase y, create another line from the lowercase s to the lowercase y. You'll see that each allele needs to have at least two connections with it. So this S has two connections with these two guys. This lowercase s has two connections with these two guys. And obviously, the uppercase y and lowercase y already have the two connections that you create with the uppercase s, lowercase s. So after creating those connections, that's what you would put at the top and at the bottom. So if we look, this one will be easier. If you create a connection between the uppercase s, uppercase y, that'll be your first row. Create a connection between the uppercase s, lowercase y, that'll be your second row. Similarly, create a connection between the lowercase s, lowercase y, that's your third row. Lowercase s, lowercase y, that's your fourth row. Another way students think about it is they use it as math. They think about it in a math term. If you label each of these alleles as numbers, let's say this is number one, number two, number three, number four. Your four combinations will be 1, 3, 1, 4, 2, 3, 2, 4. So 1, 3 in this case is uppercase s, uppercase y, as you see here. 1, 4 is uppercase s, lowercase y over here. 2, 3, lowercase s, lowercase y, and 2, 4, lowercase s, lowercase y, right here. So you may not, oh yeah, box method. So you may not need to actually create a dihybrid cross, but there have been times in the past where the question was you have a parent that's heterozygous for both traits, and you have another parent that's homozygous recessive for both traits. What is the percentage chance you'll see a heterozygous child? So in this case, obviously you'd have to create the dihybrid cross. But if you don't have, if it doesn't mention that, if both parents are heterozygous for both traits, luckily there is a ratio. So this ratio is the same thing as is similar to the one we talked about for the monohybrid cross. The monohybrid cross, it was 1 to 2 to 1. For the dihybrid cross, it's 9 to 3 to 3 to 1. You have nine children that will be dominant for both traits. Three children dominant for one, recessive for the other. Three children, vice versa, recessive for one, dominant for the other. And then one child that'll be recessive for both traits. So if there's a question with two heterozygous parents and heterozygous parents for both genes, then you can easily find the ratio because it's given to you 9 to 3 to 3 to 1. So 9 dominant dominant, 3 dominant recessive, 3 vice versa, recessive dominant, 1 recessive recessive. So, oh yeah, and then in terms of the law of independent assortment, what the law of independent assortment refers to is that genes for one trait are not inherited together with another trait. What that means is that if just because one of the gene is dominant, does not mean that your other genes associated with it will be dominant. It could be recessive, both can be recessive, etc. So different alleles, variations in alleles arise because of mutations. So random mutations can cause more than two alleles to be present in a population. However, only one individual can carry two alleles. Multiple alleles increase the number of possible phenotypes. And so a wild type allele is present the most, mutant alleles are present the least in a population. So if they ask what the wild type allele is, it'll typically be the dominant allele. Next, we're going to talk about gene interactions. So you have different types of dominance. You have complete dominance, incomplete dominance, and codominance. So in complete dominance, it's just saying whatever is dominant to the recessive gene, that is the trait that will be shown. For the top one, white is dominant to blue. So in any of the hybrids, the white color will be shown. In the second one, in this case, blue is dominant to white. So in the hybrids, the blue trait will be shown. Incomplete dominance is when neither traits are incompletely dominant relative to each other, so they actually combine. So the two characteristic traits combine. So here, in this case, you have white and blue combining to create light blue. I'm sure you've seen in your slides, they talk about like eggplants. You can have a white eggplant and a purple eggplant, and then there could be incompletely dominant to each other. So the white and purple fuse to essentially create a pink eggplant. So that's incomplete dominance. Codominance is when neither trait is similar, neither trait is dominant relative to each other. They're co-dominant. They both want to be expressed. So you'll have blue and white expression. So you have spots where they'll be, if we take the eggplant example, so a co-dominant eggplant would be if you take a white eggplant and a purple eggplant, the hybrids will be an eggplant with white and purple spots. Incomplete would be a combination, pink, and then complete would obviously be whichever one is dominant. That was the characteristic trait that will be shown. Now, polymorphism. Polymorphism is an example where a gene can have more than two alleles. It's very rare, but certain genes can have more than two alleles. So, one of the alleles is for a dark gray color. Another of the alleles is for the chinchilla color. Another of the alleles is for a Himalayan point restricted color, which is important in depending on temperature, and another of the alleles is for an albino color. So, depending on the combination of alleles, there's a specific graded series of coat color phenotype. So, in this case, you see that dark gray is dominant to the chinchilla. So, CHD stands for would be the chinchilla color. Dark gray would be dominant to the chinchilla color, but the chinchilla color is dominant compared to the lowercase c, the albino color. And then C, uppercase H, which would be your Himalayan color, which is dependent on temperature, that is also dominant to your albino lowercase c color. So a lot of these polymorphic genes, it's particularly dependent on environment, temperature, etc. So there are cases where you have genes with more than one allele, but very rare, and it would be largely dependent on temperature, environment, leading to the development of different alleles. Oh, here's the eggplant example I was talking about with incomplete dominance. Some alleles are neither dominant nor recessive, so they have an intermediate phenotype, which, as you see here, you have a pink eggplant. All right, so chapter 13, DNA and its role in heredity. So first, we're going to talk about Frederick Griffith's experiments, genetic transformation. So the transforming principle. So this is how it was found that you can have bacteria that you think is non-virulent, you think is dead, but it can come back in future generations because of a process known as transformation. So in experiment one, you have a living strain of a virus. After injecting a mouse with the living strain of the virus, the mouse dies, and you still see those living cells in the blood. And then in experiment two, you have R strain, which is non-virulent. It's just a normal strain of bacteria. You inject the mouse with it, the mouse is healthy. You don't see any bacterial cells found in the blood. If you kill the virulent S strain bacteria by heating, so kill off all the viruses, but still inject it within the mouse is still healthy. No bacterial cells are found in the blood. However, if you mix the dead cells with living non-virulent R strain bacteria, the mouse dies. So why does this happen? This happens due to something known as bacterial transformation. Bacterial transformation states that bacteria can obtain DNA from the environment and other bacteria around it. So despite this R strain being non-virulent, it can obtain the DNA from the dead S strain bacteria, it can obtain that DNA, incorporate it within itself, and become virulent instead, which is now killing the mouse, and you see living S strain cells found in the blood. So this is known as transformation. The Hershey Chase experiment was one of the most popular, incredible experience to find out what actually led to transformation. Is it the DNA being transported from bacteria to each other? Back in the day, they actually thought it was proteins being transported from one bacteria to another. So, what is the actual genetic material? So, the Hershey Chase experiment was crucial to find out and confirm what the genetic material of human life actually is. So they've used a bacteriophage known as a T2 virus to determine whether DNA or protein is the genetic material. So they labeled bacteriophage proteins with the isotope known as 35, radioactive isotope known as 35S. DNA was labeled with 32P. So depending on the virus, what would happen is that either component of the bacteriophage might be the hereditary material, and it may enter a bacterial cell to direct the assembly of new viruses. So if you find the radioactive element in uh future bacteria, that means if it was 35S, that means protein is the genetic material. If future bacteria has 32P, that means DNA is the genetic material. So after combining the virus with the bacteria and then centrifuging it, they found that the radioactive element left on the bacteriophage's DNA was only in the bacterium and not in the virus itself, which meant that DNA had entered the bacteria from the virus. So in the second experiment, when they did it with protein, they found out that the radioactive element 35S was found only in the virus. It was not found in the bacteria, which means that the protein did not go inside the bacteria, whereas the DNA actually went inside the bacteria. So that means the protein stayed on the outside, and the genetic material that actually allows for transformation and human life in general is DNA. So what is the structure of DNA? So I'm sure you guys heard ATCG is your common combination. So you have your purines and your pyrimidines. Your purines are adenosine and guanine. Your pyrimidines are thymine and cytosine. So your pyrimidines only have one ring. Your purines have two rings. So there's also something known as Chargaff's rule. Chargaff's rule is the amount of adenosine is equal to the amount of thymine, amount of guanine is equal to the amount of cytosine. Because adenine binds with thymine in DNA, guanine binds with cytosine in DNA. Also, the abundance of purines will be equal to the abundance of pyrimidines. Chargaff's rule. So uracil is also a pyrimidine. Uracil is what you find in RNA, and that's what replaces thymine when binding with adenosine. So uracil is also a pyrimidine. So there could be a question on the exam. If you have 20% adenine or adenosine, um, what will be the percentage of cytosine? So if you have 20% A, that means you also have 20% T, because A will always be forever equal to T, G will always be forever equal to T. So 20A equals 20T, that's 40% total. 100% minus 40% is 60%, which means you have 30% G, 30% C. So that's how you do the math if they ask a question like that. So A equal to T, G equal to C. Oh, I didn't go over this, but I was wondering if you guys know this. If the sequence in one strand of DNA is that sequence, what would be the complementary strand DNA? Wow, great. Everyone got it correct. Yep, the correct answer is D. In previous semesters, when I asked this question, a lot of students would select, I believe, C, but the correct answer is D because, as you mentioned, in order to find the complementary strand, you just do the opposite of whatever is in the strand. Oh, you do the opposite of whatever is in the strand. And because in this DNA, the direction is from 5 prime to 3 prime, the complementary strand would be in the opposite direction, 3 prime to 5 prime. So, yeah, great job. Everyone got it correct. The correct answer is D. Oh, and this question, I kind of went over it as well, but what's the correct answer for this question? Yep, the correct answer is 20. It was just the opposite of what I said originally. Um, all right, so structure of DNA. So Rosalind Franklin helped find the structure of DNA, which was determined through particularly X-ray crystallography. After using X-ray crystallography, it suggested that DNA was bound in a way that it formed a double helix, with the sugar phosphate backbone of each strand located on the outside of the cell. And then Watson and Crick used model building and previous knowledge of DNA to determine its official structure. And there are four key features. Number one, it is double-handed or is double-stranded. So double-stranded obviously creates that double helix. Number two, it is right-handed. So right-handed means the curve of your helix follows your direction of your fingers, whichever your thumb is in. For the most part, most DNA is right-handed. It is anti-parallel, which means one direction of the strand will be from 5 prime to 3 prime. The other direction of the strand will be from 3 prime to 5 prime. And the direction of the strand is determined by the sugar phosphate bonds. And lastly, the outer edges of your nitrogenous bases are exposed in a major and minor groove. All right. We don't need to go over this. I just went over it. It's double-stranded and anti-parallel is the structure of DNA molecules. So what is the function of genetic material? DNA helps store obviously your genetic information. It's precisely replicated in cell division, expressed as your phenotype, and importantly, it is susceptible to mutation. A lot of mutations are obviously harmful, but there are mutations that can lead to increased development. And that's how bacteria survive as well. If you think about it, if you put bacteria in an environment where majority of them will die, other than because bacteria reproduce through binary fission where each daughter cell is identical, the only way a bacteria cell will reproduce is if their genetic material mutates. So now we're going to talk about DNA replication. So what happens? DNA begins with a primer, which is a starter strand. And then the primer is complementary to your DNA template. So what happens is that primase binds to your template strand and synthesizes an RNA primer. After that RNA primer is synthesized, it's synthesized one nucleotide at a time by primase. Then DNA polymerase then adds nucleotides to the 3' end. So when the primer is complete, primase is released and DNA polymerase comes in. It's another enzyme, it comes in, it then binds to that RNA primer and starts to synthesize new DNA. So primer is taken over by DNA polymerase and it's added to the 3' end. So as you see here, you have two strands of DNA, right? You have your leading strand and your lagging strand. So the leading strand is from your 3' to 5' direction because that makes it easier to create your DNA strand from the 5' to 3' direction. Because 5' to 3' is the natural direction. So DNA polymerase will easily be able to elongate the strand if it reads the leading strand from 3' to 5'. So it creates a 5' to 3' strand. As it creates the 5' to 3' strand, you also have a lagging strand. So your lagging strand is being formed in the 3' to 5' direction, which obviously is not the greatest. So it's oriented so that exposed 3' end gets farther from the fork, while the leading strand is oriented to grow the 3' end as the fork opens. And the fork is specifically where the DNA unwinds to expose bases. So as we mentioned, the in the leading strand template, DNA polymerase is able to elongate it perfectly fine. However, for the lagging strand, it's not able to elongate it very smoothly. So how does it elongate it? It elongates it through something known as Okazaki fragments. So as it says here, synthesis of the leading strand is continuous. However, the lagging strand is synthesized as Okazaki fragments. So what happens is that there are stretches of DNA where you have multiple primers. So as you see here, primase synthesizes a primer, DNA polymerase comes in and fills in the gaps. And it'll continue on until it reaches the end of the DNA. So for leading strand, continuous, lagging strand, primase and DNA polymerase have to work together to get the lagging strand, complementary strand of DNA made. Creating these RNA primers allows gaps or creates gaps in the middle, and those gaps are filled in by DNA polymerase, and those gaps are known as Okazaki fragments. And yeah. So primus is formed here. Now there's a gap in the middle. In the lagging strand, DNA polymerase comes in and creates those Okazaki fragments. So DNA polymerase 1 replaces the primer with DNA eventually. So a DNA polymerase comes in because obviously you don't want RNA within your DNA strand. So DNA polymerase 1 eventually comes in and replaces these RNA primers with DNA. However, uh, there are gaps in the middle for Okazaki fragments. So these gaps in the middle for the Okazaki fragments are closed up by ligase. DNA ligase is known as the gluing enzyme, so that creates the final linkages between these Okazaki fragment segments so that it can be a smooth strand. So DNA polymerase 3 makes lots of mistakes in replication and DNA can be damaged in living cells. So cells have several repair mechanisms: proofreading, mismatch, and excision. So proofreading is when if bases are paired incorrectly, the nucleotide is removed and DNA polymerase adds the correct ones. For mismatch, whenever a mismatched base is detected, it's recognized so that it can excise it and get rid of it. And then for excision repair is when enzymes come in to constantly scan DNA for any damaged bases, and then DNA polymerase 1 adds the correct ones. So for mismatch repairs, it's usually scanned for mistakes by other proteins, while for excision repairs, it's when DNA is damaged by radiation, chemicals, and spontaneous chemical reactions during the life of the cell. So as you hear, this is just regular DNA proofreading. If any bases are incorrect, the nucleotide is removed so that DNA polymerase can add the correct one. For mismatch repairs, if after proofreading the DNA is still wrong, then proteins can help scan the DNA and then get rid of multiple of the bases at once so that DNA polymerase 1 can come in and add the correct ones. And lastly, during excision repair, which is when the DNA got damaged during radiation, etc., enzymes scan it, get rid of those bases so that DNA polymerase can add the correct ones. So now we're going from genes to protein. Um, oh yeah, so we'll still probably have like another half hour left just to tell you guys. Because we're gonna have to go through gene expression as well. So from gene to protein, the central dogma of gene expression, DNA gets converted into RNA through a process known as transcription, then that RNA gets translated into a protein. However, there are some expressions to the central dogma. For example, RNA can be transcribed into other RNA, and that RNA can be translated into a polypeptide. Those are known as viruses. So those some viruses use RNA, but you also have viruses that convert RNA to DNA through reverse transcription. So DNA gets transcribed into RNA, that RNA gets translated into a polypeptide, or RNA can get reverse transcripted back into DNA, and then it can become part of the cell. So translation occurs within, or transcription occurs within the nucleus itself. So the messenger RNA is formed within your nucleus. Then translation occurs where the ribosomes are, typically in the rough endoplasmic reticulum, which makes the proteins. So within the nucleus, transcription occurs, creating mRNA. Within the ribosomes, typically at the rough endoplasmic reticulum, translation occurs where protein synthesis happens. So the differences between RNA compared to DNA, RNA has uracil instead of thymine. So what's the difference between uracil and thymine? Thymine has a methyl group over here, while uracil only has a hydrogen ion. RNA also uses ribose instead of deoxyribose. If you were there in the first exam review session, I went over how DNA doesn't have an oxygen on, I believe, the second carbon or the third carbon, while ribose does have an oxygen on the second and third carbon. Uh, so because of that, deoxyribose is deoxy, does not have an oxygen. RNA binds with uracil, so your base pairings are AU and CG. And you have three types of RNA used in protein synthesis: mRNA, tRNA, and rRNA. So mRNA is your messenger RNA. So your messenger RNA is what is created within the nucleus and carries the transcript of the DNA sequence to the site of protein synthesis at your ribosome. So it is similar to your coding strand, but complementary to the template strand. And then before translation occurs, it undergoes some modifications. Next, you have your transfer RNA. So your transfer RNA is what helps translate your messenger RNA. It brings nucleic acid sequences and then adds it to create the polypeptides. You have 20 transfer RNAs, one for each amino acid, and tRNA has an anticodon that is complementary to the codon sequence on mRNA. So codons are pairs of three amino acids. Um, or not amino acids, pairs of three bases. And then each codon makes up one amino acid. So codons are three bases, and each codon codes for one amino acid. And transfer RNAs interact with mRNA and rRNA during protein synthesis. Lastly, you have your rRNA, which is your ribosomal RNA. It's transcribed constantly in your nucleoli, which helps form the bulk of ribosomes within your cytoplasm. Because you have free-floating ribosomes in your cytoplasm, and rRNA can act as structural support and act as an enzyme to help catalyze the formation of peptide bonds during translation between the amino acids. So now we're going to go over step one, which is transcription. Once again, it's in the nucleus. You have your DNA template for base pairing. You have your nucleotides and your RNA polymerase enzyme. So transcription occurs in three steps: initiation, elongation, and termination. Initiation is when you open up the DNA, bring in RNA polymerase. Elongation is adding the nucleotides to lengthen your mRNA strand. And then termination is releasing your completed mRNA. So what actually happens here? You have the promoter region. So the promoter region is a sequence of DNA nucleotides that tells your RNA polymerase, hey, come here so that you can start transcribing the RNA. So it's where the gene begins. It tells it which strand is the template for transcription and which direction to move in. So here, initiation, RNA polymerase binds to the promoter region of a gene. After RNA polymerase binds to the promoter region of a gene, it starts to unwind the DNA as well. So you see, here's the promoter. It starts to unwind the DNA until eventually it reaches the termination site. So it's reading the template strand in the 3' to 5' direction. So it's reading this strand right here, and the RNA transcript is being synthesized in the 5' to 3' direction. So the RNA transcript is anti-parallel to the template strand. So the RNA polymerase attaches to the promoter, but it's reading this bottom strand here, the 3' to 5' strand, and producing the RNA transcript in the 5' to 3' direction. So as RNA polymerase comes in and the DNA gets unwinded, it reads the 3' to 5', creates the RNA template strand until eventually it reaches the termination site. After it reaches the termination site, it's like it tells the RNA polymerase, hey, you need to stop creating this RNA transcript. So that RNA transcript leaves, and RNA polymerase either just falls off the DNA template strand, or another protein can come and push it off the template strand. So now that you have your messenger RNA, there still needs to be more work done. So this is where your post-transcriptional processing and modification occurs. So before leaving the nucleus, the messenger RNA gets cut up so that you can have only the coding sequences. So in your DNA, you have exons and introns. So your exons are your expressed regions, which are your protein coding regions. Your introns are non-coding regions and sequences. Introns are primarily there for helping with structural support. So the exons and introns, wherever they interact, those are known as splice sites. By splice sites, it means those are the sites where the exons will be cut from the introns so that in your messenger RNA, all you have are your exons. So your pre-mRNA codes for, or your pre-mRNA, the template strand is coding for everything, exons and introns. But eventually, enzymes come in. So, oh, it doesn't say here. But enzymes come in, enzymes get rid of these introns so that your actual messenger RNA is only your exons, which are your coding regions. Another thing that happens is that, so here's your coding region. Another thing that happens is a 5' GTP cap is added, as well as a polyadenosine tail. So your coding region is here. The introns are cut out. The GTP cap is added at the 5' end because it allows mRNA to bind to the ribosome. And it also helps make sure that as it's getting transported from the nucleus to the ribosome, it won't be digested by any ribonucleases. And then the polyadenosine tail is added to mature mRNA to help stabilize it. So now we're going into the actual translation portion, which, as you mentioned, is in the cytoplasm. That's where ribosomes are. And so, yeah, mature RNA travels from the nucleus to the cytoplasm. A GTP cap is added to the 5' end. Polyadenosine tail is added to the 3' end. And then two key events must happen. Obviously, codons have to be read correctly by the tRNA anticodon, and your tRNA has to bring over the correct amino acid. So here's the ribosome structure where obviously the process of creating the protein occurs. So here is your mRNA strand that the ribosome is reading. You have a large subunit and a small subunit at the bottom. So both subunits are made up of ribosomal RNA and excess proteins, and they're held together by ionic and hydrophobic forces. When these ribosomal subunits are not translating something, they exist separately from one another. So there are three sites for tRNA binding: an E site, P site, and an A site. Your E site is your exit site. That's where your amino acids will come out from. A site is your approaching site. That's where it will read the following three codons. As mentioned before, codons are three bases. So it is reading everything three codons at a time. And your P site is where the amino acid is actually brought in. So exit site, amino site, and then here tRNAs add amino acids to the peptide site. So amino acids present the next codon, so it reads the next three bases. tRNAs add amino acids to the peptide site. E is your exit site. So translation also has initiation, elongation, termination. So in initiation, you have what are known as initiation factors, which is what helps bind the small subunit to the mRNA strand. And as it binds to it, so first it binds to the 5' GTP cap, and it moves along the mRNA strand until it finds what's known as the start codon. The start codon is typically AUG. After it finds the start codon, then the large subunit comes in. So the start codon is found AUG. tRNA brings in the anticodon. The anticodon is the opposite of what's in the mRNA. So it's UAC. And tRNA brings the associated amino acid with the anticodon. So in this case, it's methionine. After it brings that associated amino acid, then the large ribosomal subunit comes in. Now peptide elongation occurs. So the large subunit helps catalyze two main reactions. It breaks the bond between the tRNA and the amino acid, and it helps catalyze the formation of the amino acid and the approaching amino acid in the amino site. So what I mean by that is now the methionine is in the P site. The A site reads the next three codons. So the next three codons that it reads in, you have a tRNA that's coming in. Your tRNA in this case brings in a PRO. It comes into the approaching site. Now, your large subunit helps break the bond between methionine and the tRNA, and it helps it create a peptide bond and bind to the proline of the incoming tRNA. So now, as you see here, you have methionine and proline, and this process will keep on occurring as the large subunit and small subunit move down the mRNA strand and more codons are read. Eventually, as it keeps going down the subunit, we started here, as it keeps going down here, eventually it reads a stop codon. As you see, more and more amino acids were added, and then the A site reads the stop codon. The stop codon in this case is UAA, and so a release factor comes in. After reading the stop codon, a release factor binds to the complex, and that release factor helps disconnect the polypeptide from the tRNA in the P site. So right now it's bound. The release factor comes into the A site. The polypeptide and the tRNA here get unbounded, and so now your tRNA and your protein chain exit through the E site. After it exits through the E site, then obviously the protein chain also exits from the tRNA as well. It disconnects the bond from the tRNA and the protein chain. And eventually, your remaining components, mRNA and ribosomal subunits, separate until eventually they come back together to code for another protein. So the job's not done though with the protein and the polypeptide chain. There are still post-translational modifications. So proteolysis, which is the cutting of a long polypeptide chain into its final products by proteases. So proteases are enzymes that can help cut it. So one gene can actually contain multiple polypeptides. So like here, this one gene can code for multiple proteins. There's also glycosylation, where adding sugars helps create glycoproteins. There's also phosphorylation. Adding phosphate groups they're catalyzed by protein kinases. Adding phosphate groups can help change the protein conformation from a tertiary structure into a quaternary structure, help it bind to other proteins, etc. And so there are different types of mutations that can occur during protein synthesis as well. So this is your wild type. This is your normal protein synthesis. So this is what will typically occur. Then you have what's also known as a silent mutation. So a silent mutation, as mentioned previously, three bases make up three bases make up an amino acid. However, there are cases where sometimes you can have different combinations of bases still code for the same amino acid. So in this case, for proline, CCC. So the combination of three C's makes proline. So three cytosines make proline. However, CCU can also make proline. So a silent mutation is when you change the sequence for one base, but you still don't have any changes in the amino acid sequence. The amino acid sequence is still remaining the same. A missense mutation is when now you're completely changing the amino acid. So here, GAU leads to asparagine. By changing the middle adenine to U, GUU produces valine. So now you have a change in amino acids. That's missense. So silent, you're still producing the same amino acid mutation despite the base change. Missense, you're producing a completely different amino acid because of the base change. Then there's nonsense mutation. Nonsense mutation is by changing one of the bases, you create a stop codon. So here UAG is a stop codon. So instead of producing UGG, instead of producing tryptophan, the tryptophan amino acid is not produced, and instead a stop codon is produced, so the polypeptide chain, no protein can be made. And lastly, there's a frame shift mutation where before previously we were talking about exchanging one base for another. In a frame shift mutation, you're adding a extra base. So in this case, you're adding an extra base here. So now everything got moved down one. So all the amino acids are changed at the point of insertion. What should have been GA here? What should have been GAG or leucine? Now it's now GAG is split apart. Now it's TGA GGC. So there's a shift in the amino acid formation. Why is the mutation in the template strand instead of the mRNA? So the mutation is in the template strand because remember the mRNA is created from the DNA template strand. So there could be slight mutations in the mRNA, but typically the mutation occurs within the DNA itself. So the DNA itself has random mutations, and then the mRNA is just the strand that reads off of that template strand and then copies it. So it just creates the complementary strand to that template strand, and then that mRNA is then used to make proteins. So that's why the mutation is in the DNA, and then the mRNA just copies whatever mutation was found in that DNA. And so all these mutations can have different functions. So, like we mentioned, there could be so the normal allele, normal transcription, normal translation occurs, you have a normal functional protein. As we mentioned before, a silent mutation, you change one of the bases, but despite the change in the base, normal translation occurs because though that different base still codes for the same amino acid, and you still have a functional protein. You have a loss of function mutation where because of the change in base, now the protein is non-functional. It lost its original function. Or what typically happens, for example, in bacteria, you can have a gain of function mutation. So in a gain of function mutation, you code for a protein with a new function. So because of that mutation, now this protein has excess functions available to it. So, all right, now we are in the last chapter. Thank you guys for staying this long. We're in the last chapter right now, regulation of gene expression. So, for gene expression, so there are several ways a cell can shut off the supply of a protein. So, downregulation of mRNA transcription. Downregulation is the process process by which a cell decreases the quantity of a cellular component, so it decreases the amount of transcription of mRNA that's occurring. Hydrolyzation of mRNA, which prevents translation, preventing mRNA translation at the ribosome, so getting rid of any binding sites, hydrolyzing the protein itself after it's made, and inhibiting the protein function. However, the most efficient of all of this is blocking the transcription in the first place. By blocking transcription in the first place, it's more efficient because imagine you transcribe the gene, translate the message, create a protein, and then you decide, no, I don't need the protein, I'll degrade it or inhibit it. So the body typically prefers to block transcription in the first place. Oh, okay. Types of gene regulation. So you have what's known as negative regulation. So you have a binding site where typically transcription would occur, but there's a repressor binding site as well, where molecules known as repressors can help bind to that protein, can help bind to the binding site. After binding to the binding site, it then prevents any transcription from occurring. So for example, common example is the lac operon. Lac operon, it'll get bound to, and then now it is repressed. So no transcription can occur, no messenger RNA is formed. There are positive regulators where typically transcription would not occur. So during positive regulation, an activator binds to the binding site. By binding to the binding site, it allows for transcription to occur. And then, so there are two ways to regulate a metabolic pathway. There's allosteric regulation and transcriptional regulation. So, allosteric regulation allows for rapid fine tuning. So, from enzyme one, you produce your end product, and the end product acts as a negative feedback. So, it inhibits the activity of just enzyme one only. So, it can quickly block the pathway. However, transcriptional regulation occurs where each individual gene is looked at and then blocked. So each individual gene, which codes for each individual enzyme, is regulated. So by regulating each individual enzyme, it helps block the transcription of all five genes so that no enzymes can be produced. So allosteric regulation is very quick, so very rapid, but transcriptional regulation allows for slower and more fine-tuned conservation of energy and proteins enzyme synthesis. So now we're going to talk about E. coli for glucose versus lactose. You guys worked on E. coli for the piglo experiment during lab. So E. coli cells like metabolizing glucose. However, if glucose is in low concentration or if there's an absence of glucose slash presence of lactose, E. coli can switch its lactose metabolism. So different genes within E. coli will start to be expressed so that it can metabolize lactose. So since E. coli doesn't like metabolizing lactose, it needs extra help. Galactoside permease is a carrier protein that becomes activated that helps move lactose into the cell. Beta galactosidase helps hydrolyze lactose, and beta galactoside transacetylase transfers acetyl groups to beta galactosides to help allow it to function. So different genes can now be expressed because of its environment. So here, having excess lactose levels in a specific environment, when lactose is added to that growth medium, the enzyme essential to the metabolism of lactose, so beta galactosidase, starts to get made. So by adding the inducer, now there's much more beta galactosidase being produced. So during this lag period, the mRNA for the enzyme is made. Now you have a specific level necessary. Because of the increased lactose in the environment, you have increased beta galactosidase. But once you remove the inducer, so in this case, once you're removing lactose from the growth medium, the amount of mRNA also decreases because now you don't need lactose anymore. The only reason you had beta galactosidase in the first place was because there was an increased amount of lactose in their environment. So this a lag period happens because obviously the body, not the body, the E. coli bacteria does not typically have beta galactosidase active for lactose metabolism because it's usually metabolizing glucose. So because of that, the mRNA needs to be translated to protein. Eventually, that protein needs to start working in. So because of that, there's a small lag period where there's like not much activity going on. But after that lag period is over, then it increases significantly until it reaches that induced level. So now we're going to talk about specifically the lac operon. So a typical operon consists of a promoter, two or more structural genes, and an operator, which is a short stretch of DNA between the promoter and the structural genes. And so some DNA sequences don't encode for any proteins, they're just the binding sites for other proteins. So as you see here, the lac operon has a promoter, operator, and it has different genes for each of the three enzymes I described previously to help translate it into protein or transcribe it into mRNA, translate it into protein, and then create it. So there are several ways to control the transcription of operons. There are inducible operons regulated by repressor proteins. There are also repressible operons regulated by repressor proteins, and the operon can itself can be regulated by an activator protein. So when lactose is absent, there is a active repressor on the operon here. So there's an active repressor preventing the binding of RNA polymerase to that area to initiate transcription because you only want to produce beta galactosidase and the other two enzymes if lactose is present in the environment. You don't want to waste energy and time producing those genes if lactose isn't present. You don't need it because E. coli typically uses glucose. So there are active repressors on the operon to make sure that RNA polymerase doesn't bind to it. And so the repressor protein itself has two binding sites, one for the operator and one for the inducer. So when there is a lot of lactose in the environment, now an inducer known as allolactase can bind to the repressor. So here, typically the repressor, as you see here, these are the two binding sites. Typically the repressor binds to this because nothing is bound to the active sites. But once the inducer comes in and binds to the active sites, the repressor becomes inactive. It separates from the lac operon so that the RNA polymerase can come in and then transcribe and eventually translate all three genes required to metabolize lactose. So in inducible systems, the inducer interacts with the regulatory protein so that the repressor can no longer bind and allows transcription. So, for example, here, the inducer binds to the repressor protein so that it can no longer bind, allow for transcription. But there's an opposite system as well. In repressible systems, a co-repressor binds to a regulatory protein, and now the repressor becomes active, binds to the operator, and blocks transcription. So inducible systems allow for transcription, repressible systems block transcription. Here it talks about the transcription differences in prokaryotes versus eukaryotes. So prokaryotes only have one RNA polymerase, eukaryotes have multiple. We have one for transcribing ribosomal RNA, two for RNA polymerase 2 helps transcribe messenger RNA, RNA polymerase 3 helps transcribe transfer RNA. Prokaryotes only have a few promoters and regulatory sequences, eukaryotes have multiple. And then for initiation of transcription, prokaryotes is when the RNA polymerase binds to the promoter, but eukaryotes RNA polymerase binds to the promoter, but there's also other proteins that are involved to either induce the system, repress the system, etc. So there's more proteins involved in eukaryotes as well. And so as a result of this, regulation can and does occur at multiple points before, during, and following transcription and translation. So here we're talking about the initiation of transcription in eukaryotes. So you have something what's known as TF2D. So TF2D is a general transcription factor, and you have something known as a Tata box. The Tata box is a large sequence of DNA where the TF2D transcription factor binds to it, and it's a promoter region. So it binds to that Tata box so that other transcription factors can follow and RNA polymerase 2 as well can bind. So RNA polymerase 2 binds after more transcription factors came in and got bound to the DNA, and now it forms the transcription complex. After forming that transcription complex, RNA polymerase is finally ready to transcribe RNA. So, like I mentioned previously, the top box is extremely important because it indicates where that genetic sequence can be read and decoded. And RNA polymerase 2 can only come in after multiple transcription factors are bound to that top box. So there are repressors and activators, etc. So one thing that's really important about DNA is DNA bending. So the reason for that is that there are sequences that bind to specific transcription factors, but they may be extremely far from the actual transcription start site. So but despite the fact that it's far from the transcription start site, you need it to bind to this transcription complex so that RNA polymerase can come in. So what happens in those cases? In those cases, DNA bending happens. During DNA bending, it allows specific transcription factors to interact with the RNA polymerase complex and affect the rate of transcription. So in this case, this is an enhancer region. It can bend so that it interacts with that transcription complex so that transcription rates can increase. And that's the benefit of DNA being a double helix. So viruses can regulate their gene expression as well. You have virus protein particles known as virions. Virions are acellular. They're usually composed of only nucleic acids and a few proteins. And viruses have to use host cells to carry out metabolic functions while they reproduce. So here you have what's known as the lytic cycle. So in the lytic cycle, the early stage is that a promoter in the viral genome, so here a promoter in the viral genome binds to a host RNA polymerase, and as a result of that, adjacent viral genes are transcribed. And then you also have late stage where viral late genes are transcribed. So these viral late genes are important for translating and producing phage proteins, the capsid, which is the protein code for the virus, and enzymes for lysis, while the transcriptions are just the virus genome itself. And so the late stage genomes are important for lysing the host cell and eventually releasing new virions. So what happens if we look at the left, the lytic cycle, a bacteriophage binds to the bacteria, gives up its DNA. Eventually, that DNA, that host DNA is digested, and then a new phage DNA forms using the nucleotides from the former host DNA. As a result of that, the host cell is able to transcribe the phage DNA and phage RNA, producing phage proteins. As you see here, it's producing the proteins, capsids, etc., to produce more virions. As it produces more virions, assembly of the phage is complete, and the enzyme known as the enzymes for lysis. So this enzyme comes in and then helps break apart that cell. As it breaks apart that cell, now new phages are released to eventually start the cycle again. However, so that's the lytic cycle. The lytic cycle is very, um, what's the word? The lytic cycle, the issue with the lytic cycle is that once the host cell, it breaks apart the host cell, so the host cell dies off, right? So it has to keep on finding new host cells because it's completely bursting apart that cell itself. However, in the lysogenic cycle, what happens is that the phage DNA, instead of digesting the host DNA to then make more virions, it actually incorporates itself into the bacterial chromosome. So it incorporates itself into the chromosome and becomes a prophage. Now that chromosome replicates itself, and this process keeps on going until eventually there's a circumstance where or environmental condition where the prophage has to excise itself out of the host DNA so that the cell enters the lytic cycle. Eventually, everything will end the lytic cycle. So here we're going to talk about control of bacteriophage lysis and lysogeny. So in a rapidly growing host, you have two proteins, CRO and CL. So the CL protein represses the lytic cycle, activates the lysogenic cycle. However, CRO activates the lytic cycle, represses the lysogenic cycle. So in a host that's rapidly growing, the CRO synthesis is low because there's enough energy to go around. Because there's enough energy to go around, the CRO synthesis is low, but the CL protein accumulates. Because the CL protein is accumulate, the bacteriophage can enter a lysogenic cycle, which means it's able to stay within the host genome and replicate because there's energy and nutrients, environmental conditions, etc., for that host cell to grow. However, eventually there will come a point where that host will not be able to grow anymore. And so that leads to stressful conditions. Under these extremely stressful conditions, Cl, which initially was high, now that protein is degraded. So more CRO protein is produced. Because more CRO protein is produced, now the genes responsible for the lytic cycle are activated, which excises, which right here, it excises the DNA from the host chromosome, and now it enters the lytic cycle. So here's an example of how genes can be activated or repressed based on the conditions. If it's in a growing host, Cl genes are activated, which allows it to be in the lysogenic cycle. However, under stressful conditions, Cl genes are repressed, CRO genes are activated so that it can enter the lytic cycle. So how does expression occur after transcription? So some control mechanisms include alternative splicing of pre-mRNA, microRNAs, translation repressors, and regulation of protein breakdown. So alternative splicing is a mechanism for generating proteins with different functions from a single genome. So as you see here, you have a DNA sequence with 6 different exons, and you transcribe everything, the exons and the introns included. However, when splicing the mRNA transcript, you can have different combinations of the exons, so you can have different proteins with different functions. So, for example, protein 1 has exons 1, 2, 4, 5, 6. It doesn't include exon 3. Protein 2 has exons 1, 3, 5, 6, does not have 2 and 4, and protein 3 does not have exon 2. Each of these 3 different proteins have completely different functions, but they're all from the same gene. Because they're from the same gene, it is much more efficient to create these protons because there's an easier, you can create multiple protons just from one single DNA transcript just by splicing and combining different exons and introns. So almost done, 3 slides left. So as mentioned previously, you have messenger RNA, microRNA, and small interfering RNAs. So microRNAs are small molecules of non-coding RNAs. They help regulate gene expression and they inhibit the translation of messenger RNAs. So, and then small interfering RNAs result in the silencing of RNAs. They typically come from viral infections and transposons, which can help. Transposons are parts of the gene which can change their location within the genome itself, and small interfering RNAs degrade specific mRNAs itself. So they are similar. However, for microRNA, proteins allow microRNA to bind to mRNA, which inhibit translation, and then that mRNA is degraded. While here for small interfering RNAs, by binding to the mRNA, they cause the degradation themselves. For translation repressors, there are multiple ways to regulate mRNA translation. So microRNAs previously or repressor proteins. When iron is low, a repressor binds to ferritin. What is ferritin? Ferritin is a protein molecule that causes an uptake of iron within the blood. So when iron is low, ferritin is repressed because you don't want more uptake of iron because it's low. You want to keep it in the blood. However, when there's a lot of iron in the blood, iron can bind to that repressor itself so that ferritin can detach, so that the repressor can be detached, ferritin can be translated so that can reduce the iron concentration in the blood. Last slide, proteasomes break down proteins. So what happens? You have a protein that's targeted for breakdown, and an enzyme comes in that attaches ubiquitin to the protein. What is ubiquitin? Ubiquitin is a sequence that allows other proteasomes to come in and recognize it. So it's sort of like a recognizing sequence. So ubiquitin attaches to the proteins, a target protein. That target protein is now recognized by a proteasome. Ubiquitin is released and recycled to target other proteins, and now that proteasome is able to hydrolyze that target protein. So that proteasome contains proteases, which are enzymes that help break down that protein. So by adding ubiquitin to protein, proteasomes are able to recognize this protein needs to be broken down, and it breaks it down through proteases. And that is the final slide. So yeah, I know that was a lot of material. Thank you everyone for being here. Please fill out the feedback form if you have any questions, we can go over anything, definitely ask away. And yeah, thank you guys for being here.